No limbs, no problem. Welcome to a video on the evolution of the snake body plan. In this video we will be looking at four main things. Firstly, background information on the evolutionary relationships between snakes and other animals. Then we'll be going on to look at elongation of the snake's body, looking at the developmental and genetic basis for it. Then we'll be looking at limblessness in snakes, specifically the genetic basis of why snakes lost their limbs. And finally, we'll be briefly looking at the impact of the body plan research has on the ecological evolution of snakes research. The genetics can get a bit complicated, but trust in me, hopefully I can explain it so that you will understand it. Now, as I said before, some background information. Snakes, along with lizards, are located in the crown group reptiles Sauria, in a group called Squamata. Sauria branches in two. You can see Squamata branches off in one direction, and in the other branch you have tortoises and turtles on Pantestodins, and crocodiles, birds and dinosaurs in Archaeosauromorpha. Ranchostophilia and Squamata together form the group Lepidosauria. Now let's look at Squamata in more detail. This order contains snakes and lizards, such as geckos and limbless lizards. Snakes are located within a group called Toxophera, shown here in red, which as well as snakes includes slowworms, chameleons and iguanas. Crown snakes are referred to as serpents, shown here in green. Debates on the origin of snakes have been going on for a very long time. One specific debate is the debate between the sister group to snakes. One hypothesis the sister group to snakes is mosasauroids, which is based on morphological evidence, which suggests the marine origins of snakes. However, this hypothesis has been criticised, and another one has been proposed, one where they're sister group to anguilliforms based on phylogenetic evidence, which suggests more of a fossorial or burrowing origin. There is no hard answer yet on the sister group to snakes, and more work needs to be done to figure it out. Let's look specifically at snakes. As said before, crown group snakes are referred to as serpents, containing all modern living snakes, seen here in pink. An example of this is the common European adder, seen in this image. Stem group snakes, seen here in blue, contain extinct lineages, such as Najash here. Together, crown group snakes and stem group snakes form total group snakes, hand serpents, also known as Ophidia. From fossil evidence, serpents' origins are traced back to the lower Cretaceous, and Ophidia as a whole is even earlier, being pushed back to the late to mid-Jurassic. As you can see on this diagram, the ghost ranges are quite large, which means divergent estimates vary, showing there is variation in divergence estimates. Bearing this in mind, we always have to remember that it is very hard to estimate things such as divergence time, because the fossil record is what? That's right, Burmese python. It's incomplete, which means as we find new fossils, they could provide new insights which could push divergence dates back even more. Now we get on to the main topic of this presentation, the evolution of the snake's body plan. Starting off with body elongation. As we know, snakes have a very long, elongated body, exemplified well in this picture of a corn snake we have here. Now let's get on to the developmental reasons behind the elongated body. The elongation of the body in snakes is achieved mainly by a dramatic increase in vertebral number. On the right we can see an X-ray of a newborn mouse and a hatching stage corn snake. The mouse shows regionalization with specialized vertebra type with a ribless lumbar region. The corn snake on the other hand shows a dramatic increase in vertebral number. 231 dorsal or thoracic vertebrae compared to only 13 in the mouse. A very large thoracic rib-bearing region running down the snake's body, as well as no ribless lumbar region. This shows a lack of regionalization compared to the mouse. The vertebrate body plan is divided up into segments along the axis of the body. Segmentation is initiated during development through the formation of segments called somites. During development, vertebrae in particular are derived from embryonic somites. These somites are formed in a process called somitogenesis. During somitogenesis, presomatic mesoderm, or PSM, see the diagram on the right, progressively segments into bilateral symmetrical epithelial somites. The segmentation happens rhythmically with periodicity matching molecular oscillators in the PSM cells. This oscillation is believed to drive the formation of new somites. The lunatic fringe gene mediates the segmentation of the somites. These oscillations happen more frequently in snakes than they do in other vertebrates, such as the mouse. This increase in oscillation rate within development time is what causes the elongation of the snake's body. Is there evidence for this? Well, yes. Here on the right are the results from an experiment by Gomez et al. The graph on the very right compares somite number between the groups tested and shows that during development, snakes have the highest amount of somites by quite some margin compared to other groups. The graph on the top shows the ratio of PSM to newly formed somites. By comparing snakes to chickens, mice and zebrafish, it shows that the snakes had up to 4 times the somite formation rate than other groups tested. The graph at the bottom shows cell cycle duration and shows that the snakes tested, the corn and house snake, had a longer cell cycle duration in the PSM than the other groups. Both these put together show that a large number of somites in the snakes is caused by an increase in oscillation rate rather than a large number of cell generations. In snakes, somitogenesis ends when the PSM shrinks, which can be seen in the far right graph where the PSM shrinks at the end. This normally occurs at the transition between the trunk and the tail of the developing snake. This is thought to be controlled by the Hox genes. Hox genes code for the transcription factors that are vital during development. Specifically in vertebrates, Hox genes are really important in normal regionalization development, such as the development of limbs and ribs. Furthermore, in vertebrates, they are clustered at 4 loci: Hox A, Hox B, Hox C, and Hox D. Yin et al. Compared snake and lizard genomes and mapped their evolutionary trajectories. They found that Hox A11, that mediates sacral formation, Hox C10, that mediates rib formation, and the aforementioned lunatic fringe gene all showed signs of relaxed selective constraints in ancestral snakes. The change in these genes may have contributed to the de-regionalization and elongation of the snake body during development. Furthermore, Hox D13, that mediates limb and digit pattern, as well as termination of somatogenesis, independently changed under relaxed selective constraints, suggesting that body elongation evolved more than once in snakes. It has been alluded to in the previous section around limb formation and Hox genes, but now we will discuss limblessness in snakes, exemplified by this Indonesian pit viper. As you can see, it has no limbs. But how? The zone of polarizing activity regulatory sequence, or ZRS, is a limb-specific enhancer of the sonic hedgehog gene. The sonic hedgehog gene regulates limb development in vertebrates. Cavon et al., seen in the far left diagram, looked at ZRS's activity during development of different animals. They found that through mutations of ZRS, it progressively lost its in vivo function as the body plan evolved from a more basal to advanced snakes. Rosito et al., shown left, looked at ZRS in limbless squamates, including snakes, and found the loss of function of ZRS in all limbless squamates shows that limblessness has many evolutionary paths. Back to Yin et al., discussed earlier, they found that 11 Hox genes were under relaxed selective constraints in ancestral snakes, and 1 gene under positive selection, Hox A9. They found evidence for the stepwise evolution of the snake body plan, with the forelimb associated Hox genes, Hox A5 and Hox A11, having been under relaxed selective constraints before the hind limb associated Hox genes, Hox C11, in ancestral snakes. This suggests a loss of forelimbs, then a loss of hind limbs. Furthermore, they concluded that hind limb loss was independent of forelimb loss, and that probably occurred more than once. Is this idea of stepwise loss of limbs supported by fossil evidence? Well, yes, it is. The stem snake Najas, mentioned earlier, this is the fossil for it, where the hind limbs are present, but no forelimbs. Right is an artistic reconstruction showing the hind limb but no forelimbs. All of this supports the stepwise loss of limb hypothesis. More fossils like this will produce new insights on the evolution of both limblessness and elongation in snakes. Now on to this brief ecology section. What can the research about body plans tell us about the origins of snakes themselves? Would they support the burrowing or marine hypothesis? Shine and Wall compared snakes to other squamates and found that the snake body plan of a long trunk but relatively short tail is more similar to burrowing lizards than it is of aquatic or surface-dwelling lizards. They suggest that this body plan reflects selection for effective burrowing in other squamate lineages. They also highlight that body elongation, as opposed to tail or neck elongation, allowed the gut volume in snakes to increase. This influences what they call a primary distinct ecological feature of living snakes, that they can eat large prey. This suggests that the elongation of the body through burrowing allows snakes to eat larger prey. Overall, these ideas from looking at the snake body plan can help us decipher the ecological origin of snakes. So we've got to our conclusion. Snakes are fascinating animals with a complex evolutionary history. The snake body plan is characterized by both elongation of the body and limblessness. We have seen that body elongation has been caused by an increase in vertebra through increased oscillation rates during somatogenesis. Furthermore, limblessness caused by mutations in ZRS, causing changes in the Sonic the Hedgehog gene expression. These changes affect limb development during the embryonic stage. Changes in the expression of the Hox gene in ancestral snakes are important in both these characteristics as well. And finally, snake body plan research can help with the debates around the ecological origins of snakes. Obviously, more research needs to be done to be able to unlock what the secrets of the snake's past. Well, that's unfortunately all we got time for today. Thank you for watching.